That's better. So now it's green.
Yeah. Okay. So for this, it wasn't on at all. So it must have just been dead battery.
There we go.
It's always something. Yeah. All right. We figured it out. For us. Okay.
So, all set?
Yep. You guys were not on the live stream. The sound was dead.
Sorry? Just the sound was
Yeah.
there. But like in person, we were not there.
The video, so they have the video,
Yeah.
but they did not have the sound.
Oh, did you tell this to Steph or?
Well, they knew,
Oh, I see.
so now they're fixing it. And it's working.
It's working now, right?
Mm-hmm.
Okay, you'll be on the live stream.
Yep.
Go be famous. Or infamous, whatever's your thing.
Exactly.
Any kind of publicity is good publicity. Just remember that.
Okay.
All right, everyone, please take your seats. We're going to get started again. So we had a great start from Tunay and John Mark, but now we're going to finish up the rest of our lecture with Christian. Take it away.
Welcome back. Okay, so let's quickly recap what we've seen in our kind of journey to map out the brain. We began first with these examples of these kind of coarse, destructive, large-scale interventions that were mostly the result of accidents that showed us that broad regions of the brain can have broad effects on personality. And then we took a more targeted approach using fMRI to ask, okay, what neighborhoods or cities of the brain are active during more specific behaviors? So which regions are involved in and responsible for creating behaviors or sensing shapes and faces? Now, however, with both of these issues, we're still missing something. Remember, the brain isn't made up of regions, it's made up of cells. And so we need to get a little bit more detail if we really want to understand what's going on in the brain. To explain why, we need to realize that brain regions are diverse. Just like cities are made up of many types of people who have different jobs and different responsibilities, so do brain regions. It's not just a single person who does, a city isn't just a single person who does the same thing, except 50,000 of them. Boston and New York have, they have lawyers, they have bankers, they have scientists, and so do brain regions. Those brain regions contain neurons that each perform very specific functions and only together kind of put together these computations. However, with fMRI, we are kind of ignoring these details, and we're really just averaging the responses between areas. And when we do that, we kind of assume that all 50,000 of these neurons, or every person in the city, is acting together to do this, to do the same thing. And when we do that, we miss all of the other functions of the individuals and the interactions and it's usually those kind of the diversity that comes together to really to create a computation. So with that in mind, our goal then should be not to figure out what the average activity of the crowd is, but to track what the individual is doing and where the individual is going and what their role is, what the individual's role is in some task, as well as how the individual is interacting with all of the other individuals. So to state this, what our goal then is to observe the activity of every individual neuron inside of our brain. And this task, especially if you think about the human brain, it's 100 billion neurons with trillions of connections and interactions between these neurons. This sounds like a task straight out of science fiction. What I'm going to show you is that it's not. We're actually beginning to realize this dream, but we're not realizing it in humans. That right now is still too complicated. If you want to think of a scientist as children learning how to read, then trying to map out the brain of humans is like a child trying to learn how to read by looking at the biggest, most complicated book that it can find, because that's what the human brain is. So if we want to learn how to read, if we're a child learning how to read, we want to find something simpler. And that's what we are doing as scientists. We need a brain that is not the most complicated, largest out there. And so to do that, scientists turn to model organisms. Model organisms are basically organisms that we use to simplify our problems, to reduce the system that we're using into something that really is kind of the bare bones. And the classic model organisms are basically, you have your mice, or usually range from your mice, to these zebrafish, to flies and yeast. And usually when you are trying to pick a model organism, you think, what is the simplest organism that I can use to answer my question? If my question is, how do cells divide? Then I need something that is a cell and is something that is dividing, which means I can go down, all the way down to yeast. However, so for our question, we need to think about what we need. So we want to map the activity of every neuron in a brain. And hopefully it's a brain kind of like ours. So in that sense, we need something that actually has a brain and has neurons. So yeast don't work for us. And again, we need something that's in a brain like ours. So invertebrates, like flies, they don't really have a brain that's organized with the same structure that we've shown you throughout the talk. So that leaves us with these two kind of classic model organisms left. And of these, the simplest, the one that we choose, is actually the zebrafish, which may seem odd. Maybe not all of you have realized that scientists use zebrafish as a classical tool for understanding the brain. But I'll kind of explain why we do. So it started in the pet store. People looking to use fish for research actually just went to the pet store to use zebrafish. And the reason for they use zebrafish is they're very easy to handle. You can kind of do whatever you want to them and they'll survive. They're very robust. But more powerful than the adult zebrafish, it's actually the larval zebrafish. The reason for that is that it's transparent. So if you want to understand how things like the intestine are developing, the heart develops, you can do that much easier in a baby zebrafish than in a mammal where one, it's developing inside of an adult mammal, and two, you can actually see through the body and actually see all of the cells as they develop. And that includes the brain. The baby zebrafish doesn't have a skull. That means that we can actually use light. Unlike in the human brain where light can't get through that skull, we can use light in the baby zebrafish brain to see it. But this doesn't do us any good, or actually more powerful, or in addition to the transparency, we also have the advantage of its size. The baby zebrafish in length is only a couple millimeters long, and its whole brain fits within a cubic millimeter, which is the size of that fMRI pixel that we saw. So relative to the human brain, it is tiny. You can't even see it if you have it at this scale. And if we kind of zoom out and compare the regions of the brain, we actually see our cortex, or frontal cortex, so we've been looking at fMRI, is a little bit truncated. It's much less well developed in the zebrafish, and that's because it's a simple organism. It doesn't do any of the complicated, it doesn't have any of the complicated emotions or abilities that humans have. But if we kind of ignore that and look at the rest of the brain, we actually see that all the rest of the fundamental regions are preserved. And this means that and this is because the zebrafish is still able to do the fundamental things that it needs to survive. It is still able to detect that it's hungry. It is still able to see its food, and it's still able to execute the behaviors it needs to grab that food. And so that means that with the zebrafish brain, we were actually able to, in theory, understand a lot of the fundamental circuits that are in place across the animal kingdom to enable these behaviors. So, okay, so let's just look straight into the zebrafish brain and see what's going on. It's still an ugly mesh. It's still 100,000 neurons. You might be able to pick out some cell bodies in here and some nuclei and say, oh, maybe that's an axon, maybe that's a dendrite. But you're going to have a fun time trying to disentangle all of this. And so we need some more tools. And we need tools in particular that somehow generate contrast within this messy mesh of wires. Tools that kind of say, okay, if I have some sort of neuron whose job I kind of know, can I pick those apart, see where they are, and see where they send their projections? Let's say, it'd be like saying, okay, can I see where all the lawyers in Harvard Square, where they are going, where are they sending, traveling to and fro from? So how do we develop such a tool? One of the ways scientists like to do this is actually to kind of look across the kingdoms of life and try to see if there are organisms that have tools that might actually help us do this. And in this case, one of the most powerful tools comes from the jellyfish, which it doesn't have a brain, but it still helps us with brain mapping. And the tool it has, kind of, you're not going to be able to see it here, but if you shut off the lights and then shine a blue light, you'll see that it actually, some of these cells, it has cells that shine green. And you might already be thinking, okay, if I can get those neurons to shine green, that might help us. But how is the jellyfish shining green? Some of you may have heard of this thing. It's our friend green fluorescent protein, GFP. And so it's a single gene that gives these jellyfish cells that ability. And so this is basically what it looks like, except zoomed many millions of times over. And the way it works is when it's just sitting there, GFP does nothing. But when you hit it with blue light, GFP gets excited and releases green light. So if we can fill up our neurons that we care about with GFP and then get some blue light onto them, then we should be able to visualize them and actually see them set apart from the rest of those messy neurons. So how do we do that? Let's think about again. Where do proteins come from? Proteins come from DNA. So that tells us that, okay, we should think about somehow changing the DNA of the zebrafish to give it instructions to build GFP in these neurons that we care about. And this is the exact approach that scientists have done to do this. We basically take 3 pieces of DNA and put them together. We take one are the instructions for actually building GFP. Then to that, we attach an address. And this isn't just like a mailing address that takes it to a specific geographical location. This is an address that specifies which type of neurons we want, the neurons with a specific job that we're interested in. And then around this, we attach something that if you were here last week, you've already seen, and that's a transposon. This is a piece of DNA that actually allows the rest of it to kind of be cut and paste into other strands of DNA, like the zebrafish genome. So then with our little circular unit of DNA, what we can do is take it and inject it into single cells, where it'll then hop into the genome, and now it's a unit that Now it's a new message and tool that the zebrafish itself has. And so if we've designed this right, then in our messy mesh of wires, we should be able to spot out these GFP-filled neurons that we're interested in. To see what this looks like, we have our zebrafish here. This is the brain with the regions highlighted. We're going to take a look at a cross-section about this size. And we have a specific type of neuron that we're interested in. These are serotonin neurons that some of you may have heard of. They do lots of things. I'm not going to go into more detail than that. And this is what it looks like. So here we've kind of reduced this area to 10 neurons at each side. And you can already see, just with 10 neurons, the kind of crazy mesh that they're forming throughout the brain, where their axons are being sent all over the place. The lights, yeah. We'll shut the lights to generate some contrast. And so we kind of spin it around. We can see where these axons are being projected all across the zebrafish brain. And that's just with 10 neurons. So, okay, so to summarize so far, we've seen that scientists have this tool, GFP, that is actually allowing us to define those individual roads that we were missing in our connectome of the human brain with fMRI. And so with this, and so in the zebrafish, as well as some other of the model organisms, we're actually now being able to build up this connectome at a much higher resolution. So, but we're still missing something, and that was what we saw in the first part of John Mark's chart. Is we still don't know what these neurons are actually doing. Why are they? What do they respond to? When are they firing? Why are they firing? So before we get to how we solve that problem, I'll take any questions. Yep. Over generation. So his question is, do brains become more different over generations? So during a generation, there is a lot of changes. So the baby zebrafish brain is much smaller than the adult zebrafish brain. Across generations, that actually depends a lot on the species you're looking at. So the parents of the zebrafish brain are going to look very similar to the offspring. However, if you can, so they'll be almost identical with some changes. If you take a more complicated animal like us, then we'll actually see very different. There'll be probably far more changes between the parent and the child. So it kind of depends on what animal you're looking at. Anything else? Yep.
Are there any studies that do comparative analysis between like the size of the brain correlating to the intelligence level?
So that's yeah. So he asked whether there are any comparative analyses that look at how the size of the brain relates to the intelligence. So more important than within the same species. So for animals, it becomes very difficult to define intelligence. There aren't any conclusive studies that say in mice where they say mice with bigger brains are more intelligent than other mice.
So one thing they looked at is the size of the social group of the animals. So how big of a group they live in. And they found that there's a correlation between how big a group you live in and how big your brain is. So that's something called the social brain hypothesis. So the reason we grew such comparatively big brains is how big they are, deal with the demands of more complicated social life.
Yeah. Yep. Yeah, so the question here is whether the lack of activity in certain parts of the brain leads to its ability to function declining. And so this kind of gets at the cellular level where connections between neurons do get weaker when they're not coactive, and they strengthen when they are active together, which has kind of been extrapolated now to this idea that when large regions of the brain are not as active, then those connections start to become weaker between neurons. I don't know of any long-term studies looking at the effects of technology. I would defer that to somebody more familiar with human brains like John Mark. But yeah, it's an interesting question that really does come down to molecular level details. Anything else before I move on? All right. Oh, sorry.
I think there's a I guess rumor or saying that even Einstein only used like 6% of his brain. Is that based on any scientific findings?
That's based off of a doctor in the 1920s who didn't know anything, and it got popular, and yeah, movies like it because then it's an easy MacGuffin, but there's no evidence for it actually being true. All right, so as I mentioned, we can figure out where these neurons are going between regions of the brain, but what are they actually doing? Our goal is to observe the activity of every individual neuron in the brain. So how do we find this activity? How do we observe the activity of a neuron? One way you might think about going about this is actually still using GFP, but changing it somehow such that its glowiness, its ability to glow when hit with blue light, is affected by whether or not the neuron it's in is firing. And this is exactly what scientists have done. We've engineered GFP to have a switch. And the switch is done by taking our GFP protein and adding a new protein to it, which we'll call CAM. Now, CAM is a calcium-dependent triggered switch. When CAM is just kind of here, stuck to GFP, and there's no calcium, which is the same calcium, by the way, that you drink with your milk, then when GFP is hit with blue light, it no longer gets excited, and green light doesn't come out. However, if calcium does come in and it kind of sticks to CAM, then the switch is flipped, such that when blue light comes in and hits GFP, it can get excited and release green light, which is cool. Okay, now we have a way to modulate the ability of GFP to glow. Green shouldn't be a verb, but how does this actually help us observe neural activity? To see how it does, let's go back to our neuron. Here it's labeled with our traditional parts, the dendrite, nucleus, and cell body, and then electricity or electric current is propagated down this axon. And what should be noted is that at rest, when it's not active, the neuron is kind of empty of positive charge, but surrounding it is a lot of positive charge, including calcium. And when the neuron fires, that calcium is actually sucked into the cell, meaning that the neuron, when it's active, is going from a state of having no calcium to having lots of calcium, which is the exact difference that we need to flip our switch. So in principle, what that means is that if we fill up our neurons, all of our neurons, or all of the neurons on the zebrafish, with GCamp, then the glowing will only take place in neurons that are firing. And in principle, we can get this mesh to kind of look like this, where neurons are popping out as they begin to fire. So this would be cool. We get a zebrafish doing lots of, and we can have its brain firing. But in order to actually learn something from this, we need the firing of these neurons to be tied somehow to some stimulus and behavior. And if you remember from fMRI, this is what they did with their imaging, is they would have an individual would go into the big magnet, and it would be shown movies, and then you could connect that activity to what they were being shown or to whatever task they were being asked to think about or perform. So this is exactly what I try to do with baby fish. So rather than asking it to interpret some poem about whether or not John has a Porsche, I take advantage of the fact that zebrafish are freshwater fish, and they don't like salt. So when I show them salt, they try to wiggle and get away. And so my question is basically, how does the brain kind of pick up that there's salt coming in and turn and communicate to other neurons that communicate to other neurons that eventually tell its muscles to wiggle away? And these fish are kind of stuck to a plate with a little bit of jello, and then I can take them into a microscope where we can shine a blue laser through it and pick up the green light that comes in through these lenses into a camera up here that allows us to record movies of all of the neurons and their activity. So if we kind of take a look at our brain again, you're going to have a side view here and the top view down here. And if we pick a small region, we can zoom in and actually look at, these are individual neurons here. So you have about 30 neurons in this little field of view. Each of these little rings is the cell body, where the dark region is actually the nucleus, which does not contain GCamp. And you can actually pick up each of the individual neurons as they're firing. I don't have this memorized, so I don't know when a firing is about to happen. But there's an active neuron, there's another one. But we're not doing this just for, like this, it looks cool, but it doesn't give us the whole picture. The whole picture is contained in the whole brain. And fortunately, this microscope actually allows us to pick up the activity across the whole brain, and giving us movies like this, where we can actually capture the dynamics of neural activity as it's being hit with salt throughout the entire brain. And you can actually put together what regions are responsible for detecting salt, and eventually, when it behaves, is this a behavior-related neuron? And so if you kind of think about, if you combine the activity profiles that you get with this, with the job, the type-specific labeling that we can get with GFP, you can start to actually piece together how the different neurons with different roles are working together to generate this whole brain activity dynamics that you're seeing here. All right. So finally, to summarize that, my part of the top, we've seen that GFP can be used to specifically label individual neurons or individual types of neurons. And then we can combine that with GCamp to actually look to label the active neurons. And so together, we can visualize this with whole brain imaging to start to piece together the way all of this is kind of working together to produce behavior. Now, this is all data that's this is something we've only had the ability to do over the last couple of years. So right now, we're kind of in this dilemma where we're getting all of this data, but there's a lot of it. We don't know what to do with it. And so it's a major now analytical problem to try to figure out what any of this means. We're now thinking about things on levels of hundreds of thousands of moving parts, and piecing that together is kind of the next frontier of where our brain mapping is going. But finally, where have we been? We've gone from accidents and kind of random lesioning to see that, to kind of try to piece together broad areas and what they might do, to actually finally being able to ask questions with fMRI about what regions of the brain might be responsible for different behaviors and sensing. And now, using model organisms and bioengineering, we're actually finally being able to piece together this puzzle at the cellular level and asking how the whole thing comes together. So, take any more questions? I think open up the floor. Yep.
Given the neuron's relationship with calcium,
Yep.
can vitamin D and calcium supplementation help normalize brain chemistry, people with depression or stuff like that?
Yeah, you do have issues when you're deficient. The brain is generally pretty good at, or your body's pretty good at kind of allocating the calcium that it has to where it needs to go. Okay. So the brain is kind of like, it usually is kind of good, but you can't really make it better by drinking more milk.
Okay.
Anything else? Oh, yeah.
So I know I'm probably jumping way ahead in studies, you guys probably won't know this yet, but so you're trying to look at an individual neuron level. Is the hypothesis that like a specific neuron is responsible for a specific part of a specific behavior over and over again for years, like even isolating it down to a fish? Like, I don't know if it's clear that question. You know what I'm trying to go with it?
Yes, so you're asking whether individual neurons across evolution might be found to be responsible for specific behaviors. Yes. Okay. Or even to the degree where at the individual, a neuron might be responsible for specific behavior. So it's very rare that you have individual neurons within an individual responsible for behavior. And zebrafish, that actually exists. And many invertebrates, that also exists, where they're actually a pair of neurons that in the zebrafish, where if they come on, then the zebrafish will perform a very specific behavior. With humans, that starts to get more complicated. The correlates to those don't really exist. But it's more what we're interested in is how kind of specific collections and constellations of neurons work together and coordinate themselves to put together behavior. Well, you still need to know the single neuron, so you know what else it's working together with to put together a behavior. I think there's one up here. Okay, so the first question was how fine is the resolution that we're getting with the imaging that we're doing? So that's in both spatial resolution as well as temporal resolution. So this spatially, we are at a resolution that's good for cells. If you want to see organelles, we can't get that. Our pixel is going to be a micron, which is about a tenth or less of the actual size of the cell. So we have good cellular resolution. We start to struggle once you get into dendrites and the kind of the finer structures. And then in time, it depends on the microscope you're using. The one that I showed here, where you're taking full volumes of the whole brain, that you're getting a full volume every second, which correlates pretty well with activity and calcium. If you want the individual firing events, you'd have to go much faster. But these give you kind of a general idea of what in that second, what was the firing rate of the neuron. So basically, how many times did it fire per second? Then his second question was, has anybody ever gone into the zebrafish and kind of cut it up into super thin slices and then use some sort of imaging technique to see where all the cells are going? So this is kind of building a straight structural connectome where you're looking at all the cells at once and kind of tracing out where each process is going for all 100,000 neurons. And this has been done. This was done in the 90s for a worm called C. elegans, but that one only has 302 neurons. So it was one guy who basically sat there for many months tracing out each cell underneath the microscope. For zebrafish, this has actually just been finished, where we have all of the images put together of every neuron at that resolution. And actually, it's like trying to be published right now, but we don't have the full connectivity where we haven't traced every single neuron. It's still like kind of the big neurons that you can see very easily and trace for a long time. Yep. That would be, yep. So the question here was first, the holy grail is to kind of get this whole brain imaging at a cellular resolution replicated in the human brain. And so the question was whether or not there have been attempts or theories about how this might be possible. And so there are theories that are very theoretical, and they actually don't even, some of them don't even involve light, but actually using DNA to encode the activity of cells, and then having each of those DNA strands have a unique barcode that can tie it back to the activity. And this is extremely theoretical. I don't know when it'll work. As for direct imaging, I think part of the problem is that light is basically out of the picture. You can't do that in a live organism, a human. And so really the next best thing is to try to keep building up with your organisms. Move on from zebrafish to mice to humans, or not to humans, but to monkeys. And there are strategies that won't be like this necessarily, but to where you can have kind of these integrated, how I did, the mice is doing this, what happened, I sacrificed the mice, and then I see which neurons were active. And so there are attempts to kind of piece together the mouse activity profiles like this. But this obviously isn't something you can do with people. But yeah, at this point, that is still science fiction, I would say.
We're going to have to wrap up that portion, but could you maybe tell us what you have down here for people to look at and people who come down and ask some more specific questions?
Yep, so what we have, so for those of you who want to kind of explore and find out more, we are going to be here to answer any of your questions. And in addition, we have a few things to demonstrate. We actually have some of the baby zebrafish here for you to look at. And in addition, we have a reconstruction of Phineas Gage's skull, so you can actually see what the damage was inside of it. And then I believe John Mark will show you some of the word map or concept map illustrations that the lab that produced that has created. So you can actually explore inside the brain to see at a better resolution what different parts of the brain are trying to do.
Let's thank the group.
So let me see, I think, is the light on?
Oh my God, they're so tiny.
Yep. It's the only way we can get their whole brain.
Those little chicken dots in here, right?
Yep, so you see the two dots? Those are their eyes. And then the rest of the body is basically completely transparent.
Oh my God. How do you pick those out?
We have like little pipettes.
Tweezers?
Well, we have suction things to kind of suck them up.
Like a eyedropper.
Exactly.
There you go. Cool.